Hello, so we are going to start a new topic. This time we are going to speak about the nervous system. So the first aspects that we are going to discuss are basic principles of the nervous system. And the very basic aspect that we can have is the separation between what we call the central and the peripheral nervous system. The central nervous system includes the brain and the spinal cord. So whenever from now on, we mention the central nervous system, we're thinking about what is in the cranium and what is in the vertebral canal. So the brain and the spinal cord. When we think about the peripheral nervous system, we are thinking about the nervous system that extends into the periphery. So we are essentially thinking about the nerves, both the spinal nerves, nerves that are connected with the spinal cord, or cranial nerves, nerves that are in great part connected with the brainstem. A different organization is seen here. So again, the nervous system, separation between peripheral and central, and within the peripheral, we can find a separate separation, the somatic and the autonomic. Somatic has to do with soma, has to do with body. So this is normally used as a synonym for voluntary. So we can talk about the somatic nervous system as being the nervous system that allows us the voluntary control over our muscles. So we are speaking about skeletal muscle control. But the somatic also includes sensations, somatosensation. In addition, we have then the other separation, the other division, that we have the autonomic or autonomous nervous system. This includes the sympathetic and the parasympathetic. The sympathetic is normally a response for fight or flight, so it's considered arousing. It's a state in which the individual needs to gather energy and resources to either fight or flight, whereas the parasympathetic is dominant in states in which people are more relaxed. Oftentimes we call this the rest and digest. Okay, so now we are going to speak about brain cells. So here we find the neuron. A neuron is the basic cell of the nervous system. It's a good time for us to look at the stereotypical or prototypical anatomy of a neuron, meaning that not all neurons will look like this, but normally neurons are represented this way, and we need to know several aspects of the microanatomy of a neuron. We are going to come back to this, but for now we can already start thinking about the anatomy of a neuron. So we have the dendrites and the cell body, which is the central portion, and then we have an axon, and the axon ends in axon terminals. There are two things represented in this picture that I would like to talk about. One is that you see that arrow over there, and the arrow over there is indicating that the direction of signals or information, if you will, so signals that convey information, goes in that direction, from the dendrites to the cell body, and from the cell body down the axon. Another aspect is that it's the size of it. So it's important that we understand that um, the axon is essentially the nerve. When we talk about a spinal nerve, for example, if we think about the sciatic nerve, which is a nerve that exits the vertebral column around the lumbar and sacral level, and it's going to go all the way to the tip of the toe. We are saying that the axons, the several axons that are that make that nerve are that long. So an axon can be relatively long. In addition to neurons, we have the glial cells. So there is a certain paradigm change as far as cells that we consider main cells and cells that are accessory or supporting cells. So in biology, there traditionally certain cells have been given much more attention, so the main cells. And then all the other cells surrounding those oftentimes were thought to be just cells that are there to support the main cells. That's a paradigm that was used for many times in the brain as well. So we have the neurons as the main cell, and we have the glial cells, which are, although more numerous, they are thought to be supporting cells. However, it's important, and we're going to develop that to a certain extent, to understand that that paradigm has been changing, and oftentimes the supporting cells don't play just a passive role of supporting the primary cells, but they actually play many roles that until very recently were unknown. But now we start to understand more and more that these cells are very important. But a picture like that for an introduction to anatomy student may be very useful. So, for example, you see the neurons represented in yellow there. Then you see the oligodendrocytes, which are wrapping around the axons, forming something called myelin. We see the microglia, and you see the astrocytes with feed processes that are contacting with a capillary. And so the author is trying to represent here the contribution that astrocytes have to what is called the blood-brain barrier. Another detail in the picture are the ependymal cells, and the author puts the ependymal cells closer to the cerebrospinal fluid. That's to indicate that ependymal cells are the main producers of cerebrospinal fluid. So the aspect, that I'm trying to constantly emphasize to you is that these pictures are not just to make the slides more beautiful or less beautiful, are very important, and you can from early on start paying attention to pictures like this, because they are going to give you a lot of information. But having said that, we are now going to the small picture, and we're going to speak about the anatomy of the neuron. So we're going to see exactly here the anatomy of the neuron. We already talked about the dendrites, the cell body, which is the nuclear center of the cell, so where the cell has the nucleus, and then an axon. But anatomically, we can think about other structures, which are more or less important, but I would like to talk with you, or at least mention those structures to you at least once. So within the same way we have a cytoplasm inside of a cell, the cytoplasm on an axon can be called exoplasm. The axon starts around the cell body, and the very initial segment is called axon hillock, which is thought to be the most sensitive area to start action potentials. Collaterals are shown when an axon branches, and then axons end in axon terminals. So we are going to speak many times in physiology about the action potential and the conduction of this action potential from down an axon. But in the neuron there are other aspects, other components that also carry to a certain degree information. They're not electrical, but they are, for example, proteins that have to travel down an axon or even up an axon, and those need filaments, and that serve the purpose, in a very simplistic way, may serve the purpose of rail tracks for proteins to move up and down, and those are called the neurofilaments or neurotubules. A neurofibril is a bundle of a neurofilament. The center of the cell is then the cell body or cell soma, and you find a lot of ribosomes because there is plenty of protein synthesis. And those ribosomes at the microscope, they called the Nissl bodies. And the perikaryon is the neuron cytoplasm. So some of these words are more important than others. I'm just trying to really give you some understanding of the basic anatomy of a neuron. We spoke about this, but I would like you to pay attention to this functional interpretation of a neuron. So the dendrites are considered to be the input. The cell body is considered to be an integration zone. And then the signal is going to be conducted down the axon. So the axon is the output of the neuron. We'll see that depending on the anatomist, an axon may it's not always the output, but that is something, a detail that I explain to you when I have the right slide. But it's more of an exception, and some anatomists will find a way around that, calling the axon, when the axon works as an input, they call it something else, but we are going to speak about that later on. And so what happens here actually are the different types of neurons. So pay attention to the first, the second, and the third neuron. So the second neuron is what you call a bipolar neuron, a neuron that is clearly one axon and one dendrite. But note that the neuron all the way to your left is what you call a multipolar neuron. So both these neurons are fairly conventional. They have the dendrites, they have the cell body, and they have axon as being a conducting zone. The one that I was speaking about, the one that is a little bit confusing for an anatomist, is the unipolar neurons. So you see there's a cell body, and seemingly we have two axons. And to avoid this confusion, some authors call only the axon the output. And to the other one, they may call it an afferent or a dendritic process. But in fairness, it is also, can be seen also as another axon. So to avoid confusion, we can do what the first group of anatomists does. Keep an axon as an output area and keep that process as being the afferent process of the neuron in the case of unipolar neurons. The myelination occurs via two different cells, depending whether we are talking about the peripheral nervous system and the central nervous system. So myelinization involves enveloping the axon in what you call sheaths of myelin. This process is fairly well established, and you can read more in the textbook about it. For us, what I would like you to know is to understand that the myelination occurs in the peripheral nervous system thanks to the Schwann cells. Whereas in the central nervous system, we have the oligodendrocytes. That's spelled out right in the first paragraph. So a myelinated neuron will look something like that, in which there are segments that have myelin and segments that are still in between those segments. There are spaces in between those segments that do not have myelin. Those are called the nodes of Ranvier. See, here you have a kind of anatomy in which originally these were called nodes of Ranvier after the anatomist that first described them. But you see the picture, more recent people are trying to avoid anatomist names and they may call it just nodes. This is okay for an anatomy textbook, but note that clinically and scientifically, most people still refer to these as nodes of Ranvier. So a myelinated axon does like this, with the nodes of Ranvier and the myelination segments. An unmyelinated axon will look much simpler. So in terms of anatomy, you find them having multiple superficial grooves. So an unmyelinated axon looks something like this. You don't have to worry about its anatomy, but what happens is that a single Schwann cell shields multiple unmyelinated axons within superficial grooves. And so this is the anatomy of unmyelinated axon. So as far as the glial cells, for a basic anatomy course, it's important to have basically one sentence for each of these cells. Then I also think I have a couple of slides just to help you understand how some of the novel roles of these cells. So here, and it could well be this slide, for example, for you. So astrocytes, oligodendrocytes, microglia, and ependymal cells. So these are roles that if you open a textbook, perhaps maybe 20 years ago, they already included these functions, or even older than that. So that's the kind of anatomy we're going for, something that is very well established. And then if you, of course, are taking a neuroscience course, you may learn more about the novel roles of these. So the astrocytes seem to be very important, as I showed you in the early picture, to help maintain the blood-brain barrier. Another aspect is that the astrocytes, along with the microglia, which is the third in that diagram, seem to participate in some sort of inflammatory process of the brain. So the astrocytes are oftentimes thought to help form scar tissue after injury. The oligodendrocytes are very important to produce myelination. The microglia have also a supporting role, which means that they do a lot, but we just don't know very well what they do. And the ependymal cells are very important to produce the cerebral spinal fluid. So you're welcome to pause here and and try to write down a simple sentence of each of these cells. In the peripheral nervous system, we spoke about the Schwann cells, which are the equivalent to the oligodendrocytes, so they are important for myelination, but also for repair. We don't speak about repair in the central nervous system to a greater degree. We speak to some repair in the spinal cord, but we don't speak about a great deal of repair in the brain. But in the peripheral nervous system, the process of repair are more established. In addition to Schwann cells, we have satellite cells. Satellite cells are relatively recently discovered, and they are important to as well as supporting. Again, see, the word supporting or supporting comes for us as a comforting word, because we know that they have some role in helping out the other cells, but it's not very clear if we can come up with a sentence that is definite. I promised you a certain, and I've been telling that these cells have some emerging roles. So here we have a synapse. So synapses are communication between neurons. And for example, this is something from 2013, not that long ago, that shows that the astrocytes are very important to basically help form synapses. And here you have the microglia, for example, as showing that if this is a weak synapse, the microglia, along with astrocytes, can destroy the synapse. So you will discuss synapses later on from the point of view of the anatomy, but also physiologically, synapses are essential for learning, essential for plasticity. And so the general idea is that, synapses are weak, so they are poorly used, they should be deleted, and synapses that are frequently used or strong, they should be stimulated. And so both the astrocytes and the microglia can have a role in those processes. So these are things that you will not find in an anatomy textbook yet. You will find in the future, because anatomy textbooks normally will just choose components that are very well established, and that's what you should really know for this course. So I wanted to cover now the basic principles of neural functional organization. And the idea is that the nervous system in general has several components. So one component is to receive sensory information and to process that information and produce motor responses. And so we have there a sensory component, which is also called afferent. I know some students say afferent, but it's actually students, it's sometimes even instructors. But if you go to a dictionary and that helps with the pronunciation, you see that the word is called afferent. And the motor component is called afferent, as in yes. What is important here to understand is that sometimes we may think motor in terms of neuroscience or neurophysiology just indicates a skeletal muscle. But note that motor indicates any response, and the responses can be given by muscle, but can also be given by glands. For example, if you have a reflex in which you produce saliva, the salivary glands need the neuron that is giving the signal for the salivary glands to produce more saliva are called motor neurons. So this is a nice slide to have an organization, and there it tells you the word synapse, which is the communication between two neurons. And see, this is an image that's very rich, so we can spend a lot of time here. And it has two more details. I'll finish just with two more details. It has the afferent, and the afferent and shows that neurons in between are called interneurons. And the last thing that I would like to mention is that you see here a unipolar neuron. The sensory is a unipolar, and see the author here call it axon, they call it a peripheral process, and an axon a central process. So different anatomies, different physiologists have a way of trying to work around with the fact that the unipolar has two axons, one being sensory and one being motor. A little bit more complicated, but still a nice picture, perhaps too ambitious now, but then perhaps less ambitious as you basically go over this at the end once we are done studying everything. You see that the kind of sensory receptors, so the afferent information, the afferent fibers, and then the afferent. See that they are afferent to the skeletal muscles, which are within the somatic, but then they are afferent to, for example, smooth muscle, cardiac muscle, adipose, and glands. Those are also motor neurons. You can distinguish, separate those that go to visceral. The viscera, remember I taught you about the word viscera, which stands for viscous, so everything that is inside from the anatomical point of view would be the things that go bad in the autopsy, because they're kind of viscous. So the smooth muscle, the glands, the cardiac muscle, and the adipose tissue. Okay, it's a little bit more complicated, but it's a nice picture to come back once you understand all those terms, and we'll cover all those. A reflex is the simplest form of behavior, so things that I can observe. And you see here a reflex that in which the afferent captures the painful stimulus, information related to the painful stimulus, passes those signals to the spinal cord. There is an interneuron involved, which synapses with a motor neuron, and the response should be a withdrawal response. This reflex, you see, involves two synapses. Some reflexes are even simpler. That's the case, for example, of what we call monosynaptic reflexes, such as the knee-jerk reflex. So we are now going to speak about basic principles of neuroanatomical organization. These are words that concepts that will be needed throughout this and the next lecture. So the first aspect has to do with the cortex. The cortex is an outer part, like the cortex of a tree. So the neural cortex is the outer part, and you see in the brain this coincides with what we call the gray matter. So when we speak about the gray matter in the brain, or speak about the cortex, it's basically the same thing. But if you look at the brain, there are also other areas of gray matter besides the cortex. So those areas are sometimes called a nucleus or a ganglia, a ganglion, plural ganglia. So what you see there are, in both of those pictures, both at the level at the higher level, but also at the brainstem, you see nuclei. Nuclei are anatomically a term that is used when, so the anatomist, to give you a better perspective, the anatomist does, when it comes to neuroanatomy, the same kind of work that has been doing for the rest of the body, trying to make sense of what we see and try to create similarities and understand a pattern, hoping to contribute to function, to the understanding of function. And so what is oftentimes in neuroanatomy done is a distinction between when we have fibers and cells that are not organized, and fibers and areas in which we have groups of cells or groups of distinct boundaries. That's the general term we use for a nucleus. A center is more of a functional term. So when we know that certain areas share the same function, we speak about a center. We can speak about a center for language, a center for motor control. And so those are the components that we see there right there. We will find, this slide doesn't tell us, but we will find that the spinal cord has a reverse organization in which the white matter is outside and the gray matter is inside. So gray matter represents essentially cell bodies, and the white matter represents the axons. Axons grouped together, they may be referred to as several names may be used to refer to groups of axons. First, each axon we can can call it a fiber. This is different from a muscle fiber. A muscle fiber is a cell. A nerve fiber is an axon. A tract is a group of axons that come from one place and go to the other place. So we have an origin and a projection. And so we can speak, for example, of a cortico-spinal tract, a tract that comes from the cortex and projects to the spine. Tracts may be organized in columns, and a column is a group of tracts. We'll speak about columns, for example, when we talk about sensory tracts. A nerve is a bundle of axons, but outside of the central nervous system. So we're talking about in the periphery, the axons are projecting to the organ, or let's not forget that the axons that are sensory, in the case of unipolar neurons, are also going into along the same nerve. So nerves oftentimes have axons that are sensory and axons that are motor, and therefore we say that a nerve like that would be a mixed nerve. Not all nerves are mixed. The final aspect that is in this picture is called a ganglion, plural ganglia. A ganglion is an anatomical collection of nuclei. So we have the cell bodies of the neurons are in a ganglion outside. You can speak about ganglia inside of the brain, but in the peripheral nervous system, that's where the cell bodies are. This is going to be very interesting, very useful for us to speak about the sensory component. So all these terms will be used, and so you will have an opportunity to see how they are used. But again, if some words come up and you don't know what they mean, you can come back to this slide, or you can just search online. Just don't immediately assume that the meaning of the word is the same as you have in everyday life. Synapses are a communication between a neuron and a neuron. Some authors like to talk about synapses, the communication between a neuron and a muscle, and a neuron and a gland. But we normally should reserve synapses for the communication between two neurons. And we will use other names for the communication, for the junction between a neuron and a muscle, that we call the neuromuscular junction. or for the communication between a neuron and a gland. That is a neuroglandular junction. See that the neuromuscular junction and the neuroglandular junction are motor, involve motor neurons, neurons that are sending out a motor signal to affect the muscle, a neuromuscular junction, or the glandular production in the case of a neuroglandular junction. Synapses will be better developed when you need to understand them, so in terms of their function. But for now, I would like you to have a few ideas about the anatomy of a chemical synapse, although we are going to speak also about electrical synapses. So synapses are junctions between two neurons. There are two types of synapses, chemical or electrical. Electrical are the ones that we are just going to mention. Use the communication between two neurons is done in a way that ions move from one neuron to the other. So they are electrical changes that pass from one neuron to the other directly, and that's why they are called electrical. The chemical synapses, the communication between the two neurons is done by a chemical substance called a neurotransmitter. So let's look at the anatomy of a chemical synapse. So the neuron that is before the synapse, so we are based on the principle of unidirectional, one direction. And so there will be a neuron before and a neuron after. The before is called presynaptic. The after is called postsynaptic. So you see here that there's electrical signal, it's called the action potential, goes down the presynaptic. There are all these vesicles and mitochondria. Those vesicles have a neurotransmitter, and upon arrival, the action potential will open calcium channels that will will trigger processes of exocytosis. So sending the vesicles with a neurotransmitter into the space between two neurons, which is called a synaptic cleft. That neurotransmitter should bind to receptors on the postsynaptic neuron. An electrical synapse has the same kind of anatomy. You can speak about a pre and a postsynaptic terminal, but the conduction is then, the communication, if you will, is not done by a neurotransmitter, but rather by ions that move across one another, move across the two neurons via gap junction channels. So whatever signal is created in the presynaptic terminal, presynaptic neuron, may move to the postsynaptic neuron thanks to the movement of the charges.